Wolfgang Seng from the University of South Dakota. Instead of in the program was Zhixiu Lu, but Liang will give the talk.
Yeah.
So his talk is predicting DILI through combined genomics indicator and ensemble machine learning approaches.
Yeah. Okay. Thank you for your kind introduction. And this talk should be delivered by my student Zhixiu Lu, but he doesn't feel a good country, so I help him deliver this talk. But basically, we get involved in the same project, and I'm his advisor, and another graduate student also gets involved in this work. And we come from the University of South Dakota, and we are part of the Biomarkers lab. And yeah, thanks for a lot of introductions this morning and afternoon section, such that I can save time. Maybe introduce why we want to use genomics biomarkers for toxicity assessment in drug development. I think the basic idea is because about 40% drug-induced liver injury cannot be detected in preclinical studies, and then we may think about genomics biomarkers will be better. And I think the underlying hypothesis is genomic biomarkers will be more sensitive than those conventional markers like pathological markers, because once the cell or the organism or the the cell or organism try to respond to the drug compound, the first thing happen is gene expression. That's why I think genomics biomarkers are more sensitive. That's why we want to use genomics biomarkers for drug safety assessment. And for this task, actually I copied from CAMDA website, and we have two tasks. One task is try to predict human clinical DILI result from cell line response, just within cell line, each cell line. And the second task is we are going to try we're going to compare the result from across cell line, which means also mentioned in a lot of other talks, in previous section presentation, like using one cell line as a training data set, and then build model, then use that model to test the result in other cell line. That's actually, we also did that. I think the big challenge for this data set, a lot of you already mentioned, probably is not, I mean, I cannot say high quality, but I think it's not balanced. It's actually unbalanced data. For example, training data set, we have 60 negative, but 30 positive. The same thing happened for validation data set. But no matter what, we try to build a better computational method, especially utilize the advantage of machine learning technique, try to make the best estimation as we can. Okay, and then first thing is we try to do the data pre-processing. Actually, these processing steps quite like others. We use Math5 to get the intensity value, and then we do the log2 transformation. And I think the difference is we use a quantile normalization for the intensity value. For log2 transformed intensity value across different arrays, and such that we can reduce the batch effect using this quantile normalization. And then finally we generate these two different data sets. One is only in CMAP expression data without considering the vehicle control. And another thing is the expression difference of the corresponding compound treatment versus the vehicle control. Here, because we already did the log transformation, so here we just get the difference instead of ratio. Okay. But no matter what, those are the two data sets we obtained, and then we design our framework. We call this framework actually basically is an ensemble framework, and ensemble based on a lot of things. First of all, we have a bootstrap sampling. Okay, from the larger data set, we try to bootstrap random sample data from this larger input data, and in our case, we set M to be 30. And then each data set has 2,000 features. Okay, and overall, on average, each feature will be sampled at least 3 times. And then next is about the feature selection. And for feature selection, we also has ensemble feature selection, because for each sampled data, we apply multiple feature selection methods. And by the end, we aggregate those features. And then next, so this kind of feature aggregation will be done independently for each sampled data set. And then next ensemble step is on the classification. So for classification, we just, because by the end, we are going to predict just for one sample, we predict one class label. So we aggregate all of them, ignore the different data set, and this classifier voting is just majority voting. Okay, that's the overall of our method. Basically, as I mentioned, there are 3 steps. In order to measure the performance of our computational work, we originally used so-called ROC curve and use AUC, area under curve, to measure how good the classification is. And then later we got the suggestion from CAMDA reviewers and asked us to also provide the metric or in other formats like accuracy, sensitivity, specificity, and MCC. So we did that later, but the tool originally designed was using ROC and AUC as a metric. Right, so first let's talk about the feature selection. A lot of previous presentation already mentioned. The feature selection actually help us identify useful biomarkers, and we hope those biomarkers will help us to link the cell response to drug toxicity. And we didn't do like the combination of features or map the feature to a higher dimension or reduce to lower dimension. We just keep the low feature, because then each feature is just genes. And later, when we identify those useful genes, we can call it biomarkers. Then because then we can take a look at annotation, and then we can know the function to a lot of things. That's why we keep as a low features. And once we identify the useful features, we call it biomarkers, we can use those biomarkers to build the learning model. I think one of the questions coming from previous talks are, what are the number of features will be going to be used for building model. And as I said, we use originally we use AUC as a metric to measure the performance of classification. So we can plot the AUCs across, versus different number of top features we selected. And then we always, maybe find an appropriate, number of features that give us the highest, AUC. And then later, actually, we thought probably this AUC can be replaced or can be add more, metrics such as, MCC, accuracy, all kind of metric can be added in and help us to identify appropriate number of, features that can be used to build, classification model. Our ensemble method is this so-called weighted, ensemble method for each, feature L. Actually, this FIL represents the rankings of feature L. by method I, okay? And then if we have from 1 to S, if we have F method, S method, then we can weight the feature, weight the ranking for that feature and get a consensus, final consensus ranking for that feature. So we did this, so-called weighted feature aggregation for every feature. By the end, we just get a final consensus ranking. I think why we want to do this feature aggregation is because different feature selection method will give you different set of features. We call it feature variabilities. But for biologists, probably biologists are more interested in so-called feature stability because they want the biomarkers that are consistently appeared in every cases. For example, every duty cases, right? They don't like the variability. But for machine learning or data mining, actually, that actually is the variability improve the classification performance. And so if we have, for example, 5 feature selection method, and we can get this Venn diagram. Here we choose top 27 features, and as you can see, only 9 are discovered by all 5 feature selection methods, which means there is a variability, right? And the 9 actually are consensus. 9 are so-called stable features. Okay. Now let's look at the performance of those feature sets. Here, each bar here represents the AUC using different feature selection methods. Those are a single feature selection method. And then usually the AUC is around 0.76. If we consider the so-called most stable feature set, that actually is the feature sitting on the center of the Venn diagram. As you can see, the performance decreased, which means stable feature set will not give you better classification performance. But if we take a look at this one, this one is the features that are discovered by at least 2 feature selection method. Okay, it has a variability, but it's at least that's each feature will be discovered by at least 2 feature selection method, and it gives us a higher AUC about 0.79, which means less stable feature set give us higher classification performance. Okay, that's why we want to do the feature aggregation and then to take into account the feature variability, and then by the end we do the ensemble. Okay, this is the ROC curve of ensemble method, and along with all other 5 independent or single feature selection method. As you can see, it's slightly ensemble method slightly is better than each single feature selection method. Definitely, this kind of evaluation steps goes through actually the cross validation. A lot of previous talks already mentioned that, right? And it's the average. Okay, it's the average result, and of tenfold cross validation, and also along with ensemble features and the ensemble classification. Okay, and I think this is a typical idea about cross validation. We divide if it's tenfold cross validation, we divide data to tenfold, and each time we just pick one as a test data, and the remaining 9-fold will be the training data set. We keep doing this 10 times such that each fold will be tested once, and the other 9 will be used as training. Okay. Now another thing I want to mention here, how good once we use 9-fold as a training data set and 9-fold as a training data set, 1-fold as a test data set. How good the classification is depends on the it's evaluated by the so-called metric. As I said, we can use the AUC. We can also use accuracy. specificity, we can also use MCC. Okay, for us, we use AUC, but actually we are going to implement or change our strategy to include more evaluation metrics into the framework. I hope you can see this result. This is a summarized result from our computational framework. And basically, the performance were measured by different metrics, like TP, true positive, TN, FP, FN. Those are the true negative, false positive, FN, and also accuracy, sensitivity, specificity, and MCC. And now it's divided the different experiment were divided to two groups. In this group actually is within cell learning and testing, which means we use, for example, use MCF7 for the training and then test MCF7 cell line, the same cell line. But for this part, actually we use one cell line as a training and then to test another cell line. So overall, we can see that prediction for positives are quite well, except one scenario, which is here, I believe. We use MCF7 as a training and to test PC3, and for true positive, it's not that good, and all others are very good, but the negatives are poorly predicted, and which actually can be told from these so-called false positives. All those false positives are from negative samples. Remember, so this is the prediction result from for validation data set, and then in that validation data set, we have in total we have 19. We have 19, negative samples, and it turns out, for some, scenario, we didn't predict any, true negative, label. I think I have some idea about this one is because, the positive means what? Means the drug has DD, effect, which means, once you treat the cell line with the drug, and the cell line has response to that drug. Okay, but negative means, drug has no DD effect. Probably cell line did not have any response. If there's no any response, then we cannot tell negative from control, which means it's probably easy to predict positive samples, but very difficult to predict negative samples. Another thought I have is probably is due to this unbalanced data set. Okay, that's actually we try to test our framework later on more balanced data or larger data set to see which case is true actually. Another conclusion we can make is so-called vehicle control actually have little effect, which you can see tell by looking at these two columns and also these two. So this is without considering the control, we just using cell line expression data itself. And this one we consider the control. Okay, we use the difference between cell line and the vehicle controls. As you can see, in terms of the performance, they are quite similar. For this MCF7, similar result we can see for the PC3 cell lines. Okay. Yeah, that actually is our conclusion about the vehicle controls. Next thing is about the cell line bias, which means if we can predict build a model on one cell line and predict the DD label for other cell lines. Actually, as you can see, we can, except this case. Okay, except this case. And this case without consider the control. If we consider control, then compare these two. Compare these two. They are quite similar to those results, just for single cell line. Okay. So if we consider controls and then cross cell line has comparable prediction as within single cell lines. That's all our conclusion. But I think the summarized result is first of all, ensemble method definitely it works. Okay, and especially because here we have a bootstrap, and then because of that bootstrap, we can divide big data to a lot of small data set. If we are going to make this tool online, then we actually the computing can be parallelized. That's actually what we did. Actually, we designed this this website, and this is an online tool, and we call it the M2ROC because originally we use a AUC on the curve to evaluate the classification performance. We might change this name because later we are going to maybe incorporate more evaluation metrics to this tool, so we might change this name. But right now it's just demo version, and it turns out runs very quickly because we parallelize the code and we use the bootstrap, and for this CAMDA data set, we can obtain the result within just several minutes. Okay, instead of training deep learning for a week or something like that, that's long. Okay. And M2ROC actually is quite open, and it can be used not only for this data set, it can be used for a lot of other applications as long as you have a larger data matrix with class labels. Even it can be used for data mining for multiple class labels, not only for just binary classification problem. Okay, I think the future work is we try to finalize this online tool and make it more efficient, and at the same time we try to integrate network analysis because here once we identify the biomarkers, a lot of you, especially biologists, are asking what are the functions of those biomarkers? Are they correlated or not? Actually, then the network analysis will help us, to answer those questions. And again, as I said, we want to include more data set for the testing and also modeling, especially more balanced data set. As I remember, this is not the first time CAMDA has dual data set. In 2013, there's another data set also try to predict duly class label using the Japanese taxol genomics data set. Actually, we also get involved in that project. It turns out the data set, at least from our experience, the result from that data set is much better than this data set. So I think probably, yeah, I think the more data set here, we will consider that data set as well. And for that data set, not only the DD has class label of 1 or 0, actually it has like drug dose level and the different treatment time. Actually it's more complicated than this data set. And right, that's actually is our opinion about this data set. Overall, we probably have the similar result as other groups. Okay, and right, so that's all I have here. And then by the end, I want to thank the CAMDA organizer and the reviewers and to give us comment, and especially the travel award to my student. And I also want to thank all my lab members. Miraj and again, Miraj and Zhixiu are directly getting involved in this project. Thank you.
Yeah, I think that most people share with you this relief with the unsuccessful performance of other groups with this data set. So it's not our fault. It seems that it's data set's fault. So there is time for a very, very short question with a yes or not answer.
Yes.
Can you?
I'll just get it off. Right. So I was just wondering, so like the recurring theme on sort of all the talks seems to have been that one of the big problems with the data set is the massive class imbalance. You know, you have about 3 times as many examples of one class as the other. So I was wondering if you looked at, so for example, you bootstrap sampled your data. Did you consider, you know, either undersampling the overrepresented class or bootstrapping to allow multiple examples from the underrepresented class to try and account for that? And then sort of in the same vein, assessing the model performance using something like a precision recall curve, which better accounts for imbalances?
No, actually, for this one, we did not. Right. Our, sampling step is just, random sampling. And so that's probably cost problem because, if the data is unbalanced, probably for each, subset or for each, sampled data, we could have more, positive samples than negative samples.
Yeah,
right?
and so especially, so a lot of people have been using random forests, which are really, really sensitive
That's
to
true.
imbalance and tend to
right.
predict whatever's more common.
Yeah. That's a good
Cool.
question, right? That's why we want to try more data sets later, or maybe we can improve the sampling, step.
Yeah.
Right.
Cool, thanks.
Okay, thank you very much. And the last speaker of this first part of the afternoon session is Maciej